Hi, and welcome to this paper where we're going to be looking at the upcoming Unified Biology paper, which you are probably going to be sitting on Friday. So what I'm going to do in this video is I'm going to give you a mock paper to do. The link is in the description below. But first, I'm going to see how did we do with our predictions, or how did I do with the predictions over the last few papers. I'm like reasonably happy with how everything turned out. This is what I kind of predicted for the processes paper. I'm not going to spend ages on this because it's, what's done is done and we shouldn't focus on what's past. But the stuff that I put in green came up quite a lot, okay? That's processes and some more processes, okay? Diversity, this is what came up, okay? So in the diversity paper, there was quite a bit on industrial fermented technology. There was loads on like bacterial growth and culture and mycoprotein and all that sort of stuff. There was a bit on globular proteins. There was some population cycle stuff. Some fish farming came up. I'm really pleased that came up. Transsex, impact of human populations on diversity, bit of gene mutations, and some transcription. So this is what I originally predicted for Unified, okay? But now we can maybe finesse this. We can like change it a little bit. So I've got some new predictions for Unified, okay? These are my new top predictions and 6-mark questions to prepare for Unified Biology, okay? So I want to go through this with you first a little bit. Okay, so the first thing we should know is that on biological processes paper, we had muscles came up. I was hoping it came up, and it did come up. So that's great. So like muscles is not going to come up again, okay? So you don't need to, well, don't forget it just yet, but it's probably not going to come up again. I still think that there might be a bit of this other stuff coming up. Controlling heart rate is, I think, maybe a practical that could come up. But there is definitely going to be some plant hormones in there. Okay, so my top predictions for this part in the Unified is uses of plant hormones commercially. I think that is going to come up in some way. And I really think that there's going to be a practical component which is about kind of measuring heart rate and doing a t-test to kind of compare heart rate before exercise and after exercise. It could be a slight difference. It could be maybe like looking at the heart rate of Daphnia water fleas before caffeine and with caffeine, and to see if there's a statistical difference using t-test. That is a practical that you may have done and might come up. But make sure you can do t-test. Make sure you know how the heart rate is controlled, and make sure you're really on it on plant hormones, okay? Okay, transport in animals, this hasn't changed. This is the same as my previous video, so I won't spend loads of time on it. There it is, exchange of fluids, the capillaries, and the stuff about electrical control of the heart, I think, is a possibility. Cloning and biotechnology. So this came up in the diversity paper that you just recently did. And it's coming up in the Unified. So what I've done here is I've kind of focused in on what didn't come up in the diversity paper. So in the diversity paper, you had some stuff on biotech for food, you had some stuff on microorganism cultures, population growth in closed cultures, and pretty much all of that, okay? So what did you not have? There wasn't much on immobilized enzymes. Now that came up like a year or two ago, so I think it's more likely that this stuff is going to come up, okay? So the cloning of animals and plants, I think, is a big area that might come up. So my top predictions are embryo splitting, advantage and disadvantage, tissue culture, therapeutic cloning, SCNT. Okay, so these questions kind of here are about that. And then communicable disease, well, that's the same. Nothing has changed there. So again, I'm not spending lots of time on it, but those are some things that you should make sure that you know how to do. Okay, so just to summarize, the changes that I've made here in terms of my predictions, top predictions to study, are really kind of, it's kind of this stuff. Okay, 1, 2, well, all this stuff, right? Plant and animal responses, basically, all that. That's kind of newish. And the cloning and biotechnology, yeah, that's kind of newish there. Okay, so I put some of these predictions into the paper, so I'm now going to jump into this video, which I already recorded a couple days ago, for the paper. So here I am. Hi there, and welcome to this Unified Biology 2022 predicted paper, Mock 2. So in this paper, I've taken some questions from old past papers. I've taken a few questions from different specs, and I've also made a few of my own questions, all based on stuff that I think could come up. So I've put quite a bit of plant responses in this paper because remember, that didn't really come up in the first paper in biological processes. There was a lot of animal response stuff, a lot of muscles, that was good, but there wasn't that much plants. So perhaps there's going to be a bit of plants in this one. And I've also made this paper, I finished this paper after you've done your biological diversity exam. So I'm going to kind of add in questions based on what has come up in that biological diversity exam or what hasn't, especially with cloning and biotechnology. Okay, so I recommend you do this paper and then come back and mark this or use this video to mark it. So you should probably do that first and then come back to the video. And if you're ready, let's get started. Okay. All right, this question is harsh. Okay, this is a hard 6 marker to begin with. I hope you enjoyed. So it says the orange tree, Citrus sinensis, is an important crop plant. Scientists put some treatments on the orange tree. They put some gibberellins on there at various stages of development. And then auxins were applied during development and to mature orange trees. Okay, so during development, various stages. All right. The treated orange trees were compared to untreated orange trees, and this is what the scientists observed. Short roots, taller, began to grow like pretty much at the same time, fewer side branches, and retain their fruit and leaves for longer. So you want to talk, it says, suggest explanations for each of these observations. So just initially, hopefully you kind of spotted this. Shorter roots, well, auxin actually inhibits root growth. Grew taller, that's, well, auxin and gibberellin, both of them. So I'll just put A plus gibberellin can cause that. All began to grow within 2 days of each other. Well, gibberellin has an effect on seeds, so we need to explain that. Fewer and shorter side branches, we're talking about apical dominance here, and the fact that auxin is the kind of thing that causes apical dominance. And retain their fruit and leaves for longer, that is, I think, gibberellins, although this is something that sometimes I forget. Okay, so let's look at the mark scheme. And it's a good mark scheme. It's a good one to potentially learn, okay? Because this mark scheme kind of tells us all the stuff we need to know about how these hormones work and what the exam board wants you to remember. So let's look at this. Okay, so the mark scheme, we'll look at the levels in a minute. But first of all, let's break it down in terms of each observation. So short roots, that's this one. Okay, so what causes short roots? Well, high auxin concentrations leads to the inhibition of root growth. Okay, that's what you need to remember. High auxin concentrations inhibits root growth. Second thing, taller plants. Well, gibberellins promote stem elongation. So I think we need to kind of remember this language. Okay, we don't just say gibberellins make plants grow taller. This is what you need to remember. So remember, promote stem elongation. Inhibit root growth up there is something we should remember. And they stimulate cell elongation and division. Remember, auxins also cause cell elongation. I think division is more gibberellins and elongation is more auxins. Just an extra thing. Growth timing. This is about gibberellins promote seed germination. So let's really try and use this language, promote seed germination. And the way they do that is they turn on enzymes. So these amylase and protease enzymes are needed to break down stores of food that exist in the seed. So seeds are kind of nutrient rich, but the nutrients in the seeds are stored away as carbohydrates or proteins, and then they're broken down by the activation of these enzymes, which produces sugars and amino acids. Quick little application, like if you make beer, you have to like malt the barley. So you have the barley seed and you allow it to germinate a little bit by putting water on it, and then it kind of sprouts. That converts the carbohydrate into sugars, and then you have sugary barley seeds, and then that's what's used to make beer. Okay, because you want the sugar and the carbohydrates. And potentially that could come up because beer, remember, comes into use of biotechnology for food. So potentially it could come up. Side branches. Auxins maintain apical dominance, and that's what you need to say. Auxins are responsible for or maintain apical dominance and inhibits the growth of lateral shoots. And then delayed fruit and leaf fall down here. It was auxin, not gibberellin. I got that wrong. So auxins slow down fruit, and auxin inhibits abscission. So abscission, like I think it sounds like scissors, right? Abscission. So abscission means cutting off the fruits or the leaves. The plant sort of cuts them off and they drop. So those are the points you could have made. How many marks did you get from 1 to 6? Well, let's look at the levels. Let's go over here to the levels. So 1 on 2. Limited number of observations included without clear links. Hopefully we could score higher than this. Level 2, 3 or 4 marks. Explanations for some of the observations with some links. So to get maximum marks, you needed to talk about every observation. So, well, most of, okay? So detailed explanations for most of the observations. Shall we say 1, 2, 3, 4, 5? So we say you had to talk about 4 out of the 5 observations to get this level 3, 5 or 6 marks. I think that's probably reasonable. And we had to say the correct hormone for each one. So short roots, auxin. Taller, gibberellins and auxin. Growth timing, gibberellins. Side branches and apical dominance, auxin. Delayed fruit fall, auxin. Okay, so pause there if you need to have more of a look. And we're going to move on. All right, 2. Okay, so this is a question on ECGs, and it says the rhythm and rate at which a human's heart beats can be determined by several factors. Figure 5.1 shows electrocardiogram traces from 2 different individuals, X and Y. Okay, here we go. Describe and explain the differences between the 2 ECGs. All right, so first of all, let's pick out some differences. Okay, so this is the QRS complex looks the same. Okay, remember this is called the QRS complex, and it looks pretty much the same as in X. Okay, X and Y. Okay, so it's not that. But what could we say that isn't there? Well, first of all, this P wave doesn't seem to be present. This is the P wave. This one before the QRS complex doesn't seem to be present in X, and also the T wave, which is to do with repolarization, is also not present. So that's weird. And then going into the explain, we kind of want to identify what this is, X, and this is all about atrial fibrillation. Right, okay, so then we want to move on to explain. So why is the cause of this? Well, basically this is atrial fibrillation up here. So you need to remember this for atrial fibrillation. So remember this QRS complex is associated with ventricular systole. Ventricular systole. So that's all good, but the atriums are all messed up. Okay, the atriums are like twitching rapidly, which is what fibrillation means. Sort of really quick, rapid, not very coordinated contraction of the atria.
Atrial fibrillation, or AF for short, is when other areas of the atria send out electrical impulses too. These irregular impulses cause the atria to twitch. When this happens, blood isn't pumped out effectively, which could cause a blood clot to form.
There's also one other thing, which I almost forgot to mention, is look at the spacing, okay? The spacing of the QRS complexes down here is very regular, but up here it's not regular. It's like 1, 2, 3, quite a big gap, 1, 2, fairly big gap, 1, 2, 3. So there's an irregular rhythm, okay? So those are all the things that you can notice. Let's look at the mark scheme. Okay, so I think I said most of these. No P phase, atrial fibrillation, idea of rapid or frequent electrical impulses in the atria. So that's kind of explaining what atrial fibrillation means, the key word, and then what that actually means. This is interesting. I'd forgotten this. So the idea of electrical impulses not only from the SAN. So the SAN hasn't taken control of the heart, and electrical impulses are being generated somewhat randomly, I guess, within the heart tissue, within the cardiac muscle, which causes an unregulated and uncontrolled twitching. Smaller gaps between the QRS phases. or you could have said a faster heart rate. This is ignore reference to T waves. I don't really know why, to be honest, but I guess it's kind of a similar mark point to P. I don't know why. Anyway, so there you go. Moving on. So the next question. An individual's cardiac output is calculated using the following equation. Cardiac output equals stroke volume times heart rate. I would remember this, by the way. Sometimes it's given. I think once or twice I've seen it not given. So I would remember it. CO equals stroke volume, SV times HR. Okay? And this just means that how much blood your heart pumps per minute is equal to the individual squish of blood. Every time your heart beats, it pushes blood out. The volume that each beat squishes out down the aorta, times the heart rate, the frequency. Okay? It's a volume times a rate equals a volume per minute. Okay, so the individual who produced ECG Y had a stroke volume of 80. Okay, so you know that that is 80. CO equals 80 times heart rate, what? So we need to find the heart rate for person Y. So that is kind of tricky. So key skill, let's look up here. So, how to do this, right? We've got time down here. So a common mistake is to only look at one beat. Okay, a common mistake is to basically look at, I'm going to zoom in here because I'm trying to peer down to make sure I get the numbers exactly accurate, which is exactly what you should do, is to look from this point to this point and to just say, well, that is one heartbeat. It is. It's more accurate to look at more, isn't it? It's more accurate to look at this point from there to this point because it's nice and clear and we can count 1, 2, 3, 4 heartbeats. Don't make the mistake of counting the peaks. 1, 2, 3, 4, 5 peaks. How many beats, how many like times round has it gone? It's once, twice, 3, 4 times. So we are here, this is at 0.25 seconds. And over here, I think it's 1 square back, isn't it? It's there. So that is, oh, hang on a minute. That's it's not 4.4. That's 4.45, yeah. That's 4.5 seconds. So what's the difference? Okay, it's a little bit laborious, but you've got to do it. What's the difference between those two times? So the 4.45 minus 0.25 equals 4.2. So 4.2 seconds. And that is 1, 2, 3, 4 beats. So how do we turn that into beats per minute? This is a bit of a numeracy thing, and I'm just going to continue to work it out here. So if there's 4.2 seconds equals 4 beats, then how many would be 1 beat? So we do divide by 4. So that would be 1.05 seconds equals 1 beat. Okay, so now the next bit is the hardest part, well, harder part numerically. So it's not a rate of 1.05 beats per minute, that's wrong. It's 1.05 seconds per beat. So to work out the rate in beats per minute, in 60 seconds, how many beats could occur? So 60 seconds divided by the time for a beat, 1.05. So the heart rate is equal to 60 divided by 1.05, which is going to be 1.05 equals 57. I'm going to keep the 0.14, 57.14, and then I've got to times that by 80. Okay, so I'm moving it down here now. So 57.14 times by 80. In fact, I'm just going to keep it on my calculator and times by 80. So then I've got a value of 4,571, and that, we've got to have units for that. It says include appropriate units. So what have I done? I've done a centimeters cubed times a beat per minute. So this is going to be a centimeters cubed per minute. Remember, it's one of the common errors. This is fine. Or you could have also given the unit as this, centimeters cubed min minus 1. Okay? but don't like mix the two. Either one of those would be the correct unit. Okay, let's just check the mark scheme. I haven't actually looked at it yet, so hopefully I got that right. Yeah, so I got, what did I get? 4571, which is allowed. Okay, so you anywhere from 4571 to 4572 and 4570. So you've got a little tiny bit of leeway. And centimeters cubed per minute. Now this says that it prefers this, so why don't you just try and use this style of unit if you can. On other information from OCR, I have seen that you can have /minute. On this particular mark scheme, it says it prefers this method. So let's try, if we can, to remember that for the next exam. Minute to the -1. Okay. All right, this is, okay, explain how the heart is controlling the activity at Y on figure 1. So like, what is Y about? So, you know, what's happening? Yeah. So this is talking about the wave of electrical activity and how it moves across the heart. So this is talking about the SAN, the wave of depolarization across the atria, delayed for a fraction of a second at the atrioventricular node, down the bundle of His, down the center of the septum, then up from the apex of the heart, up the Purkinje fibers, causing ventricular systole. So let's look at the mark scheme. And I cut out some parts of the question, so I'm just trying to find where this actual question is. Yeah, it's this one. Okay, so 3 points here. Impulse or wave of depolarization. I like to say wave of depolarization. I feel that most times, like in most mark schemes, that always gets the mark, okay? Wave of depolarization. So wave of depolarization, I've referenced the atrioventricular node. Remember, that is where it's delayed. I think it's delayed by about 0.1 seconds at the AVN before it's passed on. Then it goes down the bundle of His, and through the Purkinje fibers to cause contraction of the ventricles. Okay, great. Yeah, okay. So don't say, there's a little, sometimes the examiner notes is quite useful. Don't say signals or messages, okay? Don't just say the heart sends a signal across this and the message goes down there. Be specific. Wave of depolarization. Okay, on to the next question, which I've kind of labeled 1, but it shouldn't be 1. What should it be? It should really be question 3. Okay, so this is one of mine. So it says, the scientists Darwin, Boyson, and Jensen were key figures in working out the mechanism behind phototropism. A diagram of their experiments is shown below. In all the experiments, seedlings were, oops, that is a bad typo, were illuminated with light coming from the left. Okay, so what conclusions can be drawn from experiments 1 to 5 for 3 marks? Okay, so 1 to 5 are the experiments done by Darwin. Okay, 1, 2, 3, 4, 5. Hopefully that was clear in the question. So what can we conclude for each one? I've said this is kind of worth 3 marks. So where do I feel like you get marks if this came up? So I think that from experiments 1 and 2, we can conclude, so 1 plus 2, we conclude that the tip is necessary for the response. Tip is necessary for the response. Okay. So I think we get the mark for, I think we could probably either, if you wrote 1 and 2, tip is necessary response, that's obviously a mark, or if you wrote just experiment 2 tells us the tip is necessary for a response, that's definitely a mark. Okay, 3 and 4 we can kind of take as a pair as well. So, and for this one I think you probably do need 3 plus 4. So for this one, light is detected light is detected at the tip. So 3, if it's opaque, you know that like if the light can't reach the tip, it's not detected elsewhere. Light can't be detected down here. If the light's coming from this side, it's not going to work on the sort of stem, I guess. And if you have a transparent covering, then light can still reach the tip, and therefore it's detected. And then the plant bends. And then number 5, we could say that light, well, no, hang on a sec. So only the tip is necessary for the response. Like it doesn't seem like we need light to reach the stem also. Like if it's just on the tip, that's fine, because we can kind of block the light from the stem using this opaque shield down here, and then we still get the response. Okay, so that's kind of where I'm going for those 3 marks. First of all, the tip is necessary for the response. 3 and 4, light is detected at the tip, and experiment 5, only the tip is necessary for response. I suppose I'll be a little bit generous here as well and say that you could also get a mark for saying that 1 is for comparison. Okay, so there's 4 mark points available for the 3 marks. Okay, moving on. My next question was a 2 marker. In the Boyson-Jensen experiments, 6 and 7, the tip of the growing seedlings was cut off and then stuck back on the growing stem, separated either by a permeable gelatin layer or an impermeable layer of mica. How do their experiments provide evidence of a hormonally controlled response? So, what we need to have for this one, for the 2 marks, is, if it was a mark scheme, I would say that it's the idea that. So the idea that in number 6, chemical produced in tip can diffuse. I think we need that word diffuse. I think that's a key term that you'd have to have. Can diffuse through the gelatin. And cause the stem to bend. And then for this one, can't diffuse through the mica. So it can diffuse through the gelatin and can't diffuse through the mica. Now there's an extra bit here. I'm not quite sure how the mark scheme would really work if this was a question, but the extra thing is that the chemical is produced in the tip, but the response isn't really generated in the tip. It's not the tip itself bending, it's the chemical has to move away from the tip to slightly lower down where the response is actually generated. Okay, so if you keep this, the tip is there and the chemical, the auxin, is being produced there in the tip, but that tip doesn't bend itself, what you need to have is the auxin moving away from the tip, causing it to bend. Okay, so that means that it's a hormonal response because we have a chemical produced in one region, moving through the plant and acting at a different region. And we also know that that chemical moves, well, in this case, via diffusion, because of course plants don't have a bloodstream. Okay, so that's what I was going for there. Okay, so on to 1C. It says, using your own knowledge, explain how auxin causes seedling 1 to bend towards the light. So this is just explaining the mechanism of phototropism. So the mark scheme for that is here. So what occurs is first light causes auxin to move away. So I think that's the first thing, auxin to move away from the light is the key statement here. I'll just underline what I've written, auxin to move away. Now you might have talked about sort of like higher order explanations of this. I don't think you are required to do this, but auxins are transported away by something called phototropin proteins, or PIN proteins for short, I think. Okay, next thing that you do need to know, auxin causes pumping of H+ ions out of the cell, or out of the cell membrane, into the cell wall. So I think for this you need H+ ions, and in the cell wall, you don't necessarily have to have space, but the cell wall is a space, it is a zone. It's not a rigid thing, just a little extra on the cell wall. Remember, the cell wall is like a matrix of cellulose fibers that kind of crisscross like this. It's like a mesh. So there is a space there. This provides an optimum pH, so I think you need to reference pH and expansin enzymes. And these loosen the cell wall so that the cell can basically stretch or elongate due to osmotic pressure. Or you could have said hydrostatic pressure, okay? But remember, the plant cells, they're kind of turgid because of the difference in water potential outside and inside the cell. So water is drawn in via osmosis. So there's like a physical pressure, a hydrostatic pressure inside the cell. So if you loosen the cell wall, the cell just stretches out, causing elongation. Okay, moving on to question number 3. So this is a disease question. It says, plague is caused by the bacterium Yersinia pestis. The bacterium is a rod-shaped cell that is approximately 3 micrometers long. If you, using a light microscope with magnification of 1250, what would the length of the cell in the image produced by this microscope? Easy one. I forget how many marks this is. I think it's maybe worth 2 marks. Just check that you can still do this. So remember that we've got the actual image. Yeah, so it's got the sort of the aim triangle or the I am triangle, depending on how you want to remember it. It looks like that. So the image size is the actual times the magnification. So image equals the actual, which is 3 micrometers, times the magnification, which is 1250. So 3 times 1250 is 3 times 1250 equals 3,750. But I think we need to give our answer. I'm not sure if it tells you what you in the question. I think I may have cut off a bit here. It probably wants to give you the answer in millimeters. So this is in micrometers, so let's convert. So that's going to equal, that's a tiny image. It seems too small. 1250, yep. Yeah, that's right. So that's going to be 3.7, sorry, that's round, 3.8 millimeters. 3.8 millimeters. I'm just going to check the mark scheme because that seems a bit small, but yep, 3.75. Millimeters. Yeah. Interesting. I guess we should give it to 2dp. I'm not sure if I would get the mark for that. Maybe I'd get 1 out of 2. 3.75. Give it to 2 decimal places. It says photographs taken of the image obtained by the light microscope could be further enlarged using a projector. Why might the enlarged image be unable to tell us more about the structure of Yersinia pestis? Well, we're really looking just for a keyword here, it's resolution. So increasing the magnification of the image just by projecting it on a giant screen doesn't give you any more detail or doesn't give you more pixels, kind of. So therefore, it's resolution that's holding us back. The exam board kind of wording that we need to give is, no further resolution. It says ignore reference to further detail as it's implied in the question, but allow reference to resolution not the same as magnification. You pretty much just need to talk about resolution properly, but no further resolution would be given by projecting it on a screen. Part 3. Outbreaks of plague still occur occasionally. I think there was a recent one in Madagascar. I always talk about Madagascar, but anyway. Yeah, there was one there. Plague is transmitted by several methods, including droplet infection, close contact between people, and fleas moving between infected rats and people. Okay, suggest 2 ways to minimize the spread of an outbreak of plague. This is something that I've kind of predicted, reducing disease transmission. But there's lots of information in this question, okay? So like droplet infection, that's like coughing from one person to the next. So, masks could be used for that. Infected rats and people, like target rats. So like, I guess, rat catchers, maybe? Rat poison? Or poison? Fleas, these are insects, so insecticide. So for vector-borne diseases, the flea is really the vector here, because it's the flea bite that is generally the thing that causes plague. You can target the pathogen or you can target the vector. So the fleas you can target with the insecticide. The rats kind of like I guess the rat's kind of like a vector for the vector, if you see what I mean. So you can target the rats as well. Close contact between people, I guess you could sort of have quarantine. I'm not entirely sure I can spell quarantine, which you would really think I would be able to after 2 years, but yeah, quarantine. Let's look at the mark scheme. Let's see if I got anything. So, wear masks, increase ventilation, keep indoors, measures to exclude, not attract, or kill rats or fleas. Okay, measures to kill rats or fleas, we talked about that. Strict or immediate. I wonder if you need those words. I guess you do. It does. If it has the slash there, it needs one or the other. Strict or immediate quarantine for persons with symptoms. Allow measures to reduce overcrowding. Okay, so those are some things you could have said there. Moving on. Yeah, this one. Okay. You might have seen this question before. If you did you remember how to do it? This is a really important key question that I often go through with my students because it's quite a horrible one and it's about evaluating the claims of a company. And remember that I think maybe commercial use of plant hormones is something that maybe could come up. So this is kind of why I put this in there. So the question says, plant hormones affect the growth of plant tissues in different ways. One such effect is to promote the formation of seedless fruit. This is really what the question is about. Cytokinins are a group of plant hormones, and a commercial plant hormone firm carried out research into 3 different cytokinins. There are many different types, and they're called, these ones are kinetin, zeatin, and diatin. The firm investigated the effect of, this is important, okay? They investigated the effect of adding different volumes of cytokinin on the production of seedless fruit. The reason I've highlighted it like that, because the different volumes of cytokinin in this investigation would be an independent variable that you would change. And the production of seedless fruit would be the dependent variable that you would measure. So as the cytokinins were sprayed on the flowers, over time, the mass of seedless fruits produced by the plants was measured. Here are the results, okay? I'm going to come back to this in just a second, but let's look at the question first. The question says, on the basis of these results, the firm decided to use diatin in their new plant spray. The firm made the following claim on their packaging. Diatin is scientifically proven to cause production of seedless fruit when applied to flowers. Okay, evaluate the firm's claim using the evidence in figure 2.1. So they say diatin, scientifically proven, evaluate the claim. Right, so basically we need to look at this graph and tear it apart, okay? Because it is rubbish. These graphs are like awful. So let's look at why. Okay, first of all, do you see any numbers anywhere? No, okay, so we don't know the volumes. We've got no numbers here, here, or here. We don't have the mass of seedless fruit here, here, or here. We don't even know if the scales are the same. Does this one go from 0 to 10? Does this one go from 0 to 100? We don't even know. So there are no numbers, there's no scales. Second thing, which way round do we normally put graphs? Well, we normally have the independent variable on the x-axis and the dependent variable on the y. But these graphs are all of them, they're the wrong way round. We've got the dependent variable here and the independent variable here. So all of these things are the wrong way round. So really, like to try and understand this graph, you've got to have to like turn your head on the side and think what happens as we move this way along the independent variable. So well, this one causes a not very big increase at low levels and then causes an increase. This one quickly causes an increase, even with small levels of cytokinin. It quickly produces an increase, which then kind of plateaus. And this one, as we move along, is kind of more linear. So they've picked, which one have they picked? Diatin, which actually probably isn't the best one. I would say that zeatin would be the best one because it has a very rapid response. Okay, so that's just the data. The other thing is, there's loads of stuff we don't know. What fruit? Okay, strawberries, watermelons, like pears, who knows? What fruit is it? Another thing, what concentration? They added different volumes of cytokinin, but was the concentration here 100% cytokinin and over here 50% zeatin, etc. So there's so much stuff we don't know. This is just like an awful piece of science from this company. So I've kind of rambled on about it. Now let's look at the mark scheme and see how you did. Okay, there's like loads of things. Oh, sorry, I got so carried away that I forgot to say the important thing. Okay, let's go back because I made a mistake, which I shouldn't do. The mistake that I made is I got carried away tearing this apart, but I forgot that when you have to evaluate a claim, you do need to give one, you've got to try and find a positive, okay? You've got to try and find at least one positive, even if you think this is the worst science I've ever seen in my life, just give a positive, and then you can charge into the problems, okay? So, we should probably divide the problems, by the way, into problems with the graph and the data, and then we should also talk about maybe some problems with the method of the investigation that we don't really know about. So let's now go back to that mark scheme. All right, so first of all, the only thing that really supports the claim is that you could say that as the volume of diatom increases, the mass of seedless fruit does increase. Okay, it does. So there is some truth to that claim. However, all the things, let's do blue for data. No scale, no units, no numbers. Axes the wrong way round. Here's something I didn't spot. Error bars or standard deviation or statistical tests are not done. We have no idea, if there's no percentages, it's not percentage increase in mass. If you are measuring the increase in mass of a watermelon, you might get a large change in mass because watermelons are big, and if they go from this size to this size, that might give you an extra, 300 grams of watermelon. But that's not a huge percentage increase. If you're looking at a strawberry and it gave you the same number increase, then you'd like your strawberry go from like this to like gigantic strawberry, and that would be a much bigger percentage increase. This is always worth saying, okay? Like, throw it in there, like, all the time. Correlation is not necessarily causation. This company claimed that it's causation. You can't really be clear that one thing causes another based on this kind of simple experiment. Forgot to talk about this. It's a company. They're trying to sell their chemical, so bias, okay? If the question is about commercial use of hormones, there may be some marks for bias because companies, commercial enterprises, try to sell things. So they might be biased if they're reporting their own research. And then finally, actually, zeatin is the one that they probably should have picked. Okay, so that's all against the firm's claim. These ones down here, I'll change highlighter color to, can I pick a different color here? Okay, I'll change to pink. Is to do with the kind of validity or the method. We've got no method. We don't know the species or type of plant or fruit. We don't know anything about control variables. Did they have different soil in one case or the other? We don't know the concentration of hormone. We don't know what temperature. There's so much stuff wrong with this. Where the fruits were grown. We don't know the carbon dioxide concentration where the plants were grown. We don't know if they were grown inside or outside. We don't know the soil and the mineral content. We don't know how much water the plants were given. We don't know the light intensity of the plants received. We don't know if there were any bees pollinating the fruit. If you don't have pollination, you can't get fruit, by the way. We don't know if there were pests, weed killers, herbicides. There's no control group. There's no evidence of repeats because we don't have like standard deviation and that sort of thing. And like we don't know at this last point, I don't know, what's that? No consideration of the interaction with other hormones or processes. So basically there's so much wrong with this. It's just crazy. So hopefully you said loads of those things, okay? And actually, these sorts of things could pop up in a mark scheme asking you to evaluate a commercial use of plant hormones again. Okay, so this is some stuff to try and remember. Okay, it could come up. In terms of the marks for this question and how many did you get, it's a little bit complex. So for level 1 or 2, you basically just need to give a for and against, okay? Or you could give, right, okay, let me just break this down. Okay, so we've got validity. This is all of validity. We've got against statements, and we've got plus statement 1. Okay, so for 1 and 2, you could have plus plus minus, or you could have plus validity, or you could have, you know, all the different combinations, basically 2 of those areas, but just one statement each. Okay, for 3 and 4, you're basically looking for like plus minus minus, or plus validity minus, this kind of thing, or like plus validity, that kind of stuff. And for these up here, you need the plus, 2 validities and 2 minuses. Okay, I think that's basically what you need. So you need that to give you 5 or 6 marks. 1 positive, 2 validity statements, and 2 sort of data statements. That's going to give you the marks. Okay. A lot of people are asking me about this evaluate kind of idea. That's kind of a useful general rule of thumb. If you're evaluating an experiment and you've got data and method, this is kind of maybe what you want to remember. 1 positive thing about the data, 2 claims about the validity, that's to do with like control variables, and maybe 2 things about the data, the way it's presented, or like uncertainties through the data and stuff like that, or problems with the presentation of the data. Okay, so that was a challenging question, but I think we can get a lot out of that question and remember it for the future. Moving on. Question 5. Okay, this is one of mine. So I took part of this from an OCR-B question, which is like another type of OCR biology that you can do. And then I just kind of made the rest of it. So let's go. The student used the following method to investigate the effect of exercise on pulse rate. They wanted to test the hypothesis exercise causes an increase in heart rate. That's the hypothesis that we're testing. Let's just mention that, put that there. Okay, the student recruited members of their biology class for the experiment. The resting pulse rate was recorded for each person using the radial artery on the wrist. Okay, you don't need to know the name of the radial artery, just like that. Okay, each person ran up 4 flights of stairs at school 3 times, and the pulse rate was taken immediately after exercise. Does this sound familiar, my class? Yes, it does, because we did this. So, another student in the class said, this study will provide useful pilot data, but any conclusions drawn, made another typo there, drawn will be invalid as lots of variables weren't controlled. Give 2 examples of variables that aren't controlled in this investigation and how you might modify the investigation if it was repeated. Okay, so here are some things that aren't controlled in this investigation. Is that going to work? Yeah. So, first of all, the intensity of exercise. It's not controlled because some people might walk up the stairs and some people might run. So maybe have some sort of set time to reach the top, or a bleep test type thing would help. Or you could just replace the stairs thing and just go with running on a treadmill at a certain kilometers per hour for a certain time is a better controlled type of exercise. Physical fitness of students is not controlled. I might actually get rid of that one because I'm not really sure how you control that. I'm not sure if that's something that we need to say. In a different type of question, there might be issues around the ethics of this type of investigation, and you could, it is worth remembering that when doing an investigation like this with people and getting them to exercise, you want to make sure that everyone in the group is, able to do the investigation and is not going to be harmed in any way by doing exercise. So that is an ethical consideration, and it may be necessary for some sort of screening process to make sure that people can engage in the experiment. Okay, so the gender ratio of class, we don't really know the gender ratio of the class, I didn't say. Ideally, if we're doing a large investigation, we should have more students really than the class. The class, if you notice below, is actually only 12 students, and that includes me, because I'm in there, some of that data is me. But really we should do a bigger group of students, like 100 students, with around about a 50-50 ratio of genders. Caffeine intake, we don't know what they drank before. One student might have had 6 cups of coffee before they came to my lesson, because it was me, I did this experiment. Another one might have had none, so we could have controlled that. Food intake is a similar thing. Maybe students can eat the same breakfast at the same time. And maybe time of day affects it. I think your heart rate does actually kind of change slightly throughout the day on a normal cycle. So potentially doing this investigation at different times of day would be useful. This question, I kind of wanted to throw in there just to get you to think about the factors that affect heart rate. And I also want to show you now, just whilst we're talking about this, something else that I've just realized is quite useful. Okay, so perhaps you've done this PAG. Okay, this is a PAG which you might have done as part of your course on investigation into the effect of exercise on pulse rate. So in this PAG, you can, if you would like to, pause here and read through the procedure and the hypotheses there, and look at the method involved. There's two sort of hypotheses, null hypotheses A and B. And then there are some questions on this PAG sheet that I've given my students in the past. I don't think this year. And one of the questions is, evaluate the validity of the chosen method, the regimes used, the method provided, and the group of test subject in this investigation. So maybe this is more of an accurate way that it might be phrased in an exam. And I'll also show you the teacher sheet for this, which kind of gives an idea of what type of things you might need to say. So this, we're really looking at number 4 here, okay? So it says, I'll just highlight that for us. Can I do that? No, I can't. It's just number 4. So it says the age of the individuals, the time of day, the type and duration of exercise may have all been controlled. However, factors such as sex, weight, height, physical fitness, BMI, sustenance may not have been considered by the students. When measuring animal responses, it is difficult to account for all the factors that would cause variation. If one or both of the null hypotheses were accepted, students might suggest that a larger number of test subjects would allow for a significant difference to be identified. Okay, so I just wanted you to kind of have an understanding for all the different variables that can affect this investigation. Okay, let's move back to the question. And here it is. Okay, below it says, I'm going to delete this now so I can have more space to write. Below it says, calculate the standard deviation of the class heart rates after exercise. You may wish to use the blank columns in the table if you wish. Now, to be honest with you, I would not use the blank columns. Much better is learn how to do this on your scientific calculator. If you've got a scientific calculator, you should have a scientific calculator. Look at the model number, go to another video on YouTube and put in, standard DVF, standard STDEV, standard deviation, VPAM, whatever it is, Casio, whatever you've got. Okay, FX, I don't know what you've got, but learn how to do it, because basically if you learn how to do it, all you have to do is you just enter in all these numbers and then you press a button and it tells you it. It's quicker and you will not make a mistake and you will get the full marks. If you give the right answer, you'll get the full marks. I will show you how to do it longhand if you want to. You could skip just a minute or so, but I'm going to show you how you do it. So first of all, you have to do the x, minus the x bar, okay? And then you have to square it. So x minus x bar is the value minus the mean. Now, luckily the mean is a whole number here, so it's not going to be too laborious. So this here, I do 174 minus 154. So that's actually 20. I can do that in my head. And here you do 186 minus 154. I will just do these now, and we'll see what I get. Okay, so I've done all those. Now, I put some of them are negative, some of them are positive. I may have messed up actually and forgotten to put the sign by some of them. Like it should have been negative, but I got a positive. But actually it won't matter when you square it, because if you square a negative number, you get a positive number. So for example, 4 squared is 16. So is minus 4, okay? So that's why, mathematically, we actually square things. So I'm now going to work out this column. Okay, so now I've done this column. And by the way, the working, if this is a 3-mark question, you probably get a mark for, sort of 1 mark for each of these columns, correct? If you are doing it this way. And then the next step is to use this formula. So the standard deviation equals the square root of the sum of this over n minus 1, where n minus 1 is the number of students minus 1. So in this case, n minus 1 is going to be 12 minus 1, or 11. So the sum, we have to add all these figures. Okay, so again, this is quite laborious. It takes a long time. Much better to do in your scientific calculator. Okay, so I've got the square root of 9,440 divided by 11. So 9,440 divided by 11 equals 858. And then if I square root that, I've got 29.9. 0.3, okay? Is that right? I don't even know. Right, so it turns out that it's not. Like, I did it using Excel, which obviously you can't use in the exam, but Excel gives me a value of 28.04 for my standard deviation. So I've gone wrong somewhere. And to be honest with you, I'm not going to figure out where I did, but I have gone wrong somewhere. According to my Excel sheet, it should be 28.04. So see if you can spot where I went wrong. I've got no idea. And this just goes again to show that I think it is much better to use your calculator to do this, because every time you're punching numbers on your calculator, you introduce the chance of doing a digit wrong. And somewhere in there, I've done a digit wrong, and I've got an answer which is a bit incorrect. It may potentially be that the Excel uses a slightly different version of the formula than this version of the formula, because they are quite close. So I'm not quite sure. It could be that. But let's use this value going forwards, 28.04. Okay, then it says, then the question says, to see whether the mean values of heart rate were statistically different, the student decided to use a t-test to calculate the value of t using the formula. Calculate t, okay? So in this case, we need to take the mean of dataset 1 minus the mean of dataset 2. So that's this minus this. 72.9 minus 154. Then we divide it by this complicated sum, which is the square root of the standard deviation squared of dataset 1 over n1. Now, n1 is 12. It's the number of number of test students. So it's the standard deviation squared divided by 12. So 72.9, I'll write it out in full to make it clear. 72.9 squared over 12 plus the standard deviation for this data set here, 28.04 squared divided by 12 again. So what will we get for that? We're going to have minus 81.1 above, and the sign of t doesn't matter. So minus or positive doesn't matter. We're looking at the magnitude of the number, which is the absolute value, not whether it's positive or negative. It's like scalar or vector quantity, if you remember from GCSE. And now below, 72.9 squared divided by 12 equals, so I've got the square root of 442 plus 28.04 squared. I spotted a mistake here. I just did the calculation and I thought that sounds a bit wrong, so look at the mistakes. It's so easy to do. Goodness me. It was wrong there, wasn't it? 72.9. It should have been 14.2. Okay. Thankfully, because I've written it out, I have spotted my mistake. So I'm going to do that again. So that's 16.8. Okay, so minus 81 divided by that, so let's work this out, 16.8 plus 65.5. And then square root that. Oh my God. 9.07, so it's minus 81.1 over 9.07, which gives me an ultimate value of t of 8.94. That was super long. Okay, well, I got there. 8.94 or thereabouts is the value of t. Did you get something roughly similar? Hopefully exactly the same, maybe a couple decimal places out depending on rounding. So key points from that: know the maths, be able to do it. I really suspect that you are going to have to do a t-test calculation somewhere. Perhaps not t-test and standard deviation, but one of the two I think is going to come up in unified. So know the maths for both of these. Okay. Moving on, what does this mean? Is it significant or not? It says, using the table of degrees of freedom, determine whether the student should accept or reject the null hypothesis. Exercise has no effect on heart rate. That is the null hypothesis. So the first thing I need you to do is work out the degrees of freedom. So in this investigation, the degrees of freedom is equal to the number of tests. So it's like the number of subjects in test 1 minus 1 plus the number of subjects in test 2 minus 1. So it's 12 minus 1 plus 12 minus 1, or 24 minus 2, which is equal to 22. Okay, so the degrees of freedom I'm looking at is 22. Now I purposely did this in this table because 22 actually isn't on this thing here. But what can we say? We can say that if we use 20 for degrees of freedom, or if we use 25 for degrees of freedom, this value 8.94 is still way bigger than either of those two things. So 8.94. So for the answer we say the calculated value of t is larger than the critical value. We could say at 22 degrees of freedom. And we could even say, maybe in brackets, because it's larger than the critical value at 20 degrees of freedom. We can see that the critical value is kind of getting bigger as we go up here. So if it's bigger than the critical value at 20, it's if it's bigger than the critical value for 20 degrees of freedom, then it's definitely bigger for the critical value at 22 degrees of freedom. So, and then we say, therefore, we reject the null hypothesis. And you have to say it like this. Therefore, we reject the null hypothesis. The difference, I'm rushing here, sorry, my writing is terrible. The difference is significant and is not due to chance. Okay, and if you say all of those things, you're guaranteed to get marks. I mean, that could have been a 3 marker, and I've said all the things that it could have been there. Okay, bigger than the critical value, reject the null hypothesis, it is significant and is not due to chance. Okay, moving on. And actually I realized when I'm making this video that I forgot to put in a question. I meant to put this in the paper, but I kind of forgot to write it in for some reason. So I'm throwing in this extra question now in the video. It says, extra question here. How would the increase in heart rate be triggered in someone doing exercise? Okay, so just maybe pause the video here and see if you can just say out loud 4 points for this. This could be a 3 or 4 mark question. Pause and have a go. Okay, well here it is. Here are the answers. So remember, we have stretch receptors in the muscles that pick up the fact that the body is moving. We also have chemoreceptors that pick up a decrease in blood pH caused by increased CO2 from cellular respiration. We could go into more detail there. CO2 from cellular respiration reacts with water to form carbonic acid. That carbonic acid dissociates into H plus ions and HCO3 minus ions, and those H plus ions decrease the blood pH ever so slightly. The cardiovascular center in the hypothalamus is going to take that information and kind of integrate it, and then it's going to send an electrical impulse, don't say message, an electrical impulse or action potential down to the sinoatrial node in the heart via the accelerans nerve, and that's going to basically increase the frequency with which the heart beats. Okay? All right, I will just get rid of that now. Okay, question 6. Right, this one I threw in because, as you know, in the last paper, processes, they threw in a 6 marker which was not on the advanced information. So who knows, they might do it again. So I put this one in about excretion, but it is also still about tissue fluid, okay? So the question is this: the process of ultrafiltration in the kidney, that's excretion, shares similarities with the formation of tissue fluid. That is transport in animals. That is on the advanced information. Describe the similarities and differences between ultrafiltration and the formation of tissue fluid. So this kind of thing could come up in the paper because, like, I guess if it's 6 marks, you could kind of say 3 marks of it is like excretion and 3 marks of it is from transport in animals. So maybe. Okay, so how did you do on this question? I've taught for my class a kind of specific focus on this exact question, so I'm just going to jump into my PowerPoint on that task, okay? So this was the question. It says the process of, there it is, it's the same question. So it's a versus question. You need to think in your head comparatively between the two processes. And here's what the examiner said about this sort of similarities and differences question. It says here, hang on one second, it says over here, those candidates who took the lead from the question organized their answer into similarities and differences. That's what I would like you to do. Okay, and those who sort of jumped around or just sort of talked for like a paragraph about ultrafiltration and then talked for a paragraph about tissue fluid, they didn't do so well. So this is kind of how you could prepare this question. And this, if you have time, and you're wanting to prepare a compare and contrast type question, this is how you can do it. Take some pictures and do similarities and differences. And I'll just take you straight to the list of similarities and differences, okay? So similarities. So both, and when you're marking your answer, if you're doing that now, you should just like write S1 for this if you got this one, S2 for this if you got this one. So how many did you get? So small molecules are filtered in both scenarios, in ultrafiltration and in tissue fluid. Both involve capillaries. Large molecules remain in the blood in both processes. There is a high hydrostatic pressure in both processes. Many molecules are reabsorbed back into the capillaries, like down over here. There's some reabsorption going on. Blood vessels become narrower to maintain pressure. I mean, it is credited. I don't think we really go in that detail with capillaries normally, so I'd be impressed if anyone got that. Hydrostatic pressure is greater than oncotic pressure in both. 6. White blood cells can't pass through. That's 7. Both involve, and this we don't really talk about, it's not in the textbook. In fact, the textbook doesn't say anything about basement membrane around capillary, at least the textbook that I use, but maybe yours does. So again, I'd be impressed if you got that. So those are all the similarities. Differences. So where does the filtrate go? Over here, the filtrate goes into the sort of, it just becomes tissue fluid and it bathes the cells. Whereas over here, it goes into the Bowman's capsule. So here it says enters the Bowman's capsule, whereas over here, bathes the cells, or intercellular space is what you could call that. And over here, and another difference, molecules that are not reabsorbed by capillaries form urine, but over here, if it's not reabsorbed, it goes to lymph. So Bowman's capsule versus intercellular space, urine versus lymph. And over here, 3-layer filtration, and over here, just 1-layer filtration. Ultrafiltration is a 3-layer. You've got the, what is it? It's the epithelium, sorry, the capillary endothelium. And then we've got the podocytes, and we've also got the basement membrane. And then over here, the last one is we've kind of got a knot of capillaries, or a bundle glomerulus of capillaries, whereas in tissue fluid, you just got like a capillary just running straight. Okay? So those are the marks that you could have got. And here's an example of a 5-mark answer. Okay, so if you wanted to pause, the writing's quite difficult, but this is a real 5-mark answer that the exam board has given us there. I'll briefly show you the mark scheme, although I think we've kind of gone into quite a lot of detail there. The mark scheme for this question is, in terms of the levels, says this. I'll just show you the top 2, okay, because that's what we're really focusing on. So for 5 to 6, correctly describe similarities and differences between the processes. And, I would say that we really need probably, to really get 6, we're going to say 3 similarities and 3 differences. Maybe for 5, you could have 2 and 3. So actually, this is fairly easy to mark. It's pretty much just count them up. And this says a similarity and a difference. Okay, so you just get 1 and 1, but well described. Okay, so let's move on. Okay, on to question 7. It says, outline the process that leads to the production of antibodies against an unfamiliar bacterium. And then I'm actually going to kind of look at 7a, part 1 and 2 together. So part 2 says, explain how T helper cells act to speed up these processes. So I've put a picture over here, which is going to help quickly run through it, and then I'll show you the mark scheme in a minute. Okay, so let's follow the stages of this process. So a macrophage, could be a macrophage or it could be a neutrophil, any phagocyte kind of eats the unfamiliar virus or bacteria. Okay, so for bacteria, it's the same thing. The macrophage would just eat the bacteria. When that bacteria is taken in, it's taken into the phagolysosome, and it's broken down. Remember, sort of digestive enzymes are kind of put into the phagolysosome, and then the pathogen is broken up into tiny little pieces. Now, some of those pieces get placed on special proteins on the surface of the macrophage. You might learn about those at university. If you go on to study pathology, they're called the MHC proteins. And it's like a little pedestal. So the macrophage then goes off to a few different places in the body. It carries with it the antigens that it's put on its surface, and it goes and presents them. Now, it can present them to 2 different things. It can present them to T lymphocytes, or in fact, it can also present them to B lymphocytes. And these lymphocytes, T or B, initially we call them naive, okay? So these naive B cells and naive T cells are just waiting, okay? They're there right now in your body, okay? They're waiting either in the thymus or in the bone marrow, is it? I can't remember. Anyway, they're waiting in places for a macrophage to come along. And they will only be stimulated if the macrophage presents an antigen that exactly matches their receptor. Okay, so that's this receptor protein here. So if that receptor protein, I'll zoom in, matches that antigen that has been presented. So on this little diagram here, this thing there is the antigen that's been presented, and it exactly matches the receptor. If it's a match, then the naive T cell or the naive B cell is activated. Okay, so that activation causes differentiation and proliferation. Okay, so proliferation means it copies itself. We call that clonal expansion. So this matching part here is called clonal selection, selecting the right naive T lymphocyte or naive B lymphocyte. And then the clonal expansion is the copying by mitosis of those naive T cells and B cells. Okay, let's look at the B cells first. The B cells, they activate, they undergo clonal expansion, and then they would differentiate into plasma cells. So plasma cells would produce the specific antibodies in copious quantities, thousands and thousands and thousands, like I think every minute or even every second, like loads. Okay, now those antibodies would help the immune system. Remember also that when in that process of differentiation, some of the cells differentiate, in fact most of them, into plasma, but some of them would differentiate into memory cells to stick around for next time. Okay, so that's B cells. The next part of the question that I asked you was about T cells. So T cells can go into a couple different types of way. They can either go into killer T cells, which wasn't the focus of this question, or they could go into helper cells. And T helper cells really just help other processes, as you might expect. So they can secrete cytokines to help this process of B cell differentiation. And they can also secrete cytokines to encourage more phagocytosis. So they can also kind of help this process over here. Okay, T helper cells. And those, incidentally, are the ones that are targeted by the HIV virus. They sort of go into and kill T helper cells, which messes up the immune system. Okay, so let's look at the mark scheme for this question now. Okay, remember it's 2 parts. First of all, kind of what's going on to produce antibodies, and then the role of T helpers. Okay, there's lots of mark points because it is a very complicated process. So the first mark point up here is B cells or lymphocytes have this antigen receptor on the surface that is only complementary to one antigen. I think I should have said that word complementary, okay? I'm not sure if I did. So it's complementary to one antigen. I'll highlight each mark point in a different color because it's quite wordy. This is then selected and activated, and it proliferates or clones or divides by mitosis. So this would be, I think you would get marks for saying clonal selection and clonal expansion occur. Then it differentiates into a plasma cell. The plasma cell is the one that secretes antibodies. I'll go back to yellow. Secretes antibodies specific or complementary to the antigen. Okay, so let's quickly recap. So antigen is complementary to the receptor on the surface of the naive lymphocyte. That naive lymphocyte undergoes clonal selection and clonal expansion by mitosis. It differentiates into a plasma cell, and then that secretes antibodies. Okay, the T helper bit down here. So I didn't mention this. So the helper T cells are themselves stimulated by antigen-presenting cells, and then they release cytokines. Okay, release cytokines, and these themselves can stimulate B cell proliferation, mitosis, or clonal expansion. Okay, so tricky question there about specific immunity, and that is something which I think might come up in this paper, specific immunity. Moving on. Okay, figure B, 16.1, shows the concentration of new antibodies in the blood of a person infected for the first time by a pathogen on day 0. This is their primary response. Okay, so this is the primary response. Then it says, on day 30, this individual was again infected with the same pathogen. Sketch a line on figure 16.1 to show the antibody concentration from day 30 afterwards. Now, I remember that last time I set this question for a class, there were some people who didn't spot this on day 30, and basically they were a bit sloppy, and they just put their line from here going straight up. But the person has not been reinfected at that point. So basically, we need to start at day 30 or 31, you know, pretty quickly, going up, okay? And the thing about it is, it's got to be going up rapidly. So it has to be going up with a steeper gradient than it did first time. First time there was a lag, and then it was a bit up. This time it should maybe, you know, go up really steep, okay? Super steep, and then it's going to reach a higher level. Exactly where, I don't know, but somewhere around there. And the other thing about the second time you are exposed to an antigen, the antibody levels remain higher for longer. So we're kind of looking for a sort of longer downslope. Something like that, okay, for 2 marks. Oops, what have I just done? There we go. Okay, then it says, explain how memory cells cause the difference between the 2 lines on the graph. I'm a bit zoomed in here, let me zoom out. So, memory cells, remember, remain in the blood, you know, after the initial infection. So they remain in the blood here. And what you need to say about the memory cells is memory cells can, if exposed to the pathogen again, they can much more quickly differentiate back into B lymphocytes and back into plasma cells very rapidly. Okay, so we can have faster production of antibodies. Okay, let's look at the mark scheme for those 2 questions, the graph and the memory cell thing. So for the graph, for the first mark, we need a higher peak and a steeper rise, okay? Higher and steeper. The line has to leave the x-axis somewhere between day 30 and 33, not before 30 and not after 33. And the concentration at 60 days has to be above peak of above the peak of the printed line. Okay, it has to be basically higher than the first response. Like, but it should really be significantly higher, not just a little bit, but like, you know, significantly higher. So for the memory cells thing, why is it faster and, you know, why is it a bigger response? So the first time round, you get a mark here for actually saying the memory cells are not present in the primary response, but they remain in the blood after the primary response. They're still there. So there is faster clonal selection. Yeah, faster, you could maybe say faster clonal selection and faster clonal expansion. All right, moving on. Okay, question 8. This figure represents an influenza virus. Various protein antigens are attached to the outer surface of the virus. I kind of threw in this question because there is a good chance that they kind of throw in something about coronavirus in this paper. I mean, why not? It's like a really topical scientific issue. So there might be something about coronavirus in this paper. And this question is kind of similar because it's about how viruses change over time. It says, when a virus infects a human host, it causes the host cells to produce many new copies of the virus. The vaccination must be given each year because there are frequent mutations in the RNA of the virus. Okay, this is exactly the same thing as with coronavirus. Okay, there are frequent mutations in the coronavirus, which gives us, Omicron, Delta, Alpha was before that. Now we've got like B4.5 or something. So there's constantly new types. The antigens on the surface of the virus are made of protein. The virus uses the organelles and enzymes to produce new copies of itself, suggests the role of viral RNA in the production of viral proteins. Okay, so this is kind of transcription and translation really. So for RNA, remember RNA goes off to the ribosome and controls the order where amino acids are joined together at the ribosome. Okay, so this is the kind of thing you wanted to say. Maybe you could talk about tRNAs and pairing codon and anticodon, but it's 2 marks, so I don't think you would need to go into too much detail. You could also maybe talk about endoplasmic reticulum and that sort of stuff, but let's have a look at the mark scheme. Okay, yeah. So viral RNA acts as messenger RNA. Sometimes I forget to say the obvious thing. So the viral RNA acts as the messenger RNA. It carries the code or the sequence to the ribosomes. Yeah, I mean, this is why I kind of did this thing myself. If you're looking at the examiner comments over here, it says candidates often could not express the difference between viral RNA and host mRNA. Yeah, they are different, so you just have to say that. Okay, moving on. Explain why mutation in the viral RNA leads to a change in the 3D shape of the protein antigens. Okay, so this is a kind of classic mutation question. So if we have a change in the RNA, we have a change in the sequence of bases, we have a change in the codons on the viral RNA. This will cause a different anticodon in the tRNA to match to that codon. Therefore, a different amino acid will be brought in by the ribosome and joined to the growing polypeptide chain. The different amino acid might have a different R group, which might be hydrophobic, where it used to be hydrophilic, and that kind of thing. That will affect the bonding. That will affect the primary structure, the secondary structure, the tertiary structure of the protein, and the folding of that protein, which will give it a different shape. Okay, so that's a lot of stuff there for 3 marks. There's loads of things you could have said. Mark scheme looks like this. Okay, so altered base sequence changes the primary structure. R groups are different. The R groups interact differently. So the tertiary structure would be, which would be there, will be different. The 3D shape is different. So it says, accept implication that 3D is tertiary structure. And I think that's it. Okay, so that was a 3-mark question. Potentially if it was longer, you might have got also marks talking about codons and anticodons and the ribosome and kind of how it works as well. Okay, moving on. The head teacher of the school decided to offer teachers free vaccinations every year. Suggests why the head teacher thought this would be a good use of the school's money. My school does this, and it is a good use of money because it means that we don't get sick as much, us teachers. So that means that the school doesn't basically have to pay for supply teachers. Supply teachers cost a lot of money, by the way. It costs like a couple £100 a day for the school to hire them. So here are the answers. Money would be saved, education improved, fewer sick days, all this stuff, okay? So if you look at this here on my paper, it says insert cloning and biotech questions here. So I'm going to pause for a day. It's currently Thursday when I'm recording this. I'm going to look at your paper, which you do on Friday, so I can kind of target the questions. Because if it comes up in diversity, the cloning and biotech stuff comes up in the biodiversity paper, it's probably not going to come up exactly the same in unified, is it? It'll be some different style of question. So I'm going to finish this video over the weekend or maybe even on Friday and then post it after your diversity paper. Okay, on to the last question, which is on embryo splitting. So embryo splitting is a technique often used by livestock farmers. A basic diagram of this process is given below. Now note that you probably wouldn't get a diagram like this in an exam. I'm just kind of putting it in there to help you learn the process, okay? And give you a little bit of a helping hand. So you should kind of know this diagram in your mind. It should just be there. My question says, a young farmer learns about the technique and makes the following claim. I'm going to clone my best cow 100 times using this technique to give me the best possible herd of cattle. There's no way this can go wrong. Evaluate the farmer's claim. Pointing out errors, areas of agreement, and possible problems were he to pursue this strategy. Okay? So there's a few things here, okay? First of all, because we've got sperm and egg, we wouldn't actually be able to clone a cow in this technique. What we do is we would take a high-value cow to get the egg and a high-value bull, and then we'd mix the two. Well, we mix the sperm and the egg from the two in a dish, get fertilization in vitro, and then we get the embryo. The embryo, which is here, produced from fertilization, is not a clone of either parent. It has got 50% of the genes from the bull, 50% of the genes from the cow. But then we do some splitting. Okay, we split the embryo using a very fine scalpel, and like you actually kind of suck the embryo onto this little platform, and then you get a scalpel and finally slice it. And then you get two different embryos, which will then grow and develop into kind of a ball of cells, which you can then implant into a surrogate mother. Okay, so let's look at a potential Mart scheme for this. Okay, so here's my kind of idea for a Mart scheme, and this is a bit of a weird question, to be honest, but I think if you tackled this question, it will give you a good understanding of all the issues and possible ways that they could assess your understanding of this, okay? So first of all, I'm going to drop in a picture of the embryo cloning process over here to help you kind of see if you understood it. And here's some of the points. Okay, so first of all, things that the farmer said that were inaccurate. Okay, so the cows produced would not be a clone of either parent. Okay, they are produced by basically in vitro fertilization and the fusion of two gametes. So in this process, we get mixing of genes. So the farmer would need to choose a bull with desirable characteristics and a cow with desirable characteristics to breed together. For example, these characteristics might be, if it's a dairy farmer, might be high milk yield for the cow, and for the bull, remember bulls do not produce milk. So like if you're looking for the desirable characteristics of the bull, how do you know if they're going to produce, like if they have good milk genetics? You have to look at their history of the daughters that they've produced. So that, like a track record of producing daughters with a high milk yield. But you could say that the embryos produced would be genetically identical to each other, but not to the parents, okay? The one thing the farmer said that is accurate is the farmer would be able to produce many cows with desirable characteristics quickly using this technique, maybe more than you could get with traditional breeding. So problems, he says there's no way this can go wrong. Problems, the farmer's herd would be of low genetic biodiversity and all his cattle would be equally susceptible to disease. So for example, okay, so problems, the farmer's herd would be of a very low genetic biodiversity. They'd all be they'd be clones. And all his cattle would be therefore equally susceptible to any disease that came along. So for example, if foot of mouth disease came along, I think that infects cattle, then like he could potentially lose his whole herd. A better strategy might be to produce some of his herd with this strategy and to split at least 3 embryos from different bulls and cows. This is a bit of a weird suggestion, but what I'm guessing is like maybe only produce 50% of your herd using this strategy, and of those 50%, maybe do, you know, a bull and a cow and split that embryo a few times, a bull and a cow and split that embryo a few times, and a bull and a cow and split that embryo a few times, so that you've got, you know, genetic diversity built into the herd, okay? So that is the end of this paper. I don't actually know if that adds up to 70. It's more or less adding up to 70. So that's kind of the length of the paper. But what I recommend that you do is also go back and look at the biological processes predicted paper and revisit any questions about plant hormones, because that hasn't really come up very much yet. And you might also want to go back and look at the biological diversity predicted paper and revisit any questions about animal and plant cloning, because that hasn't come up yet, okay? Okay, so that's probably the last video from me before your Unified Biology paper. So if this is the last biology video that you ever watched, thank you for spending it with me. I really hope that lots of you are going to go study biology at university and continue being interested in biology. There's loads of really good YouTube channels that are less exam focused, which I hope you explore, and I wish you the best of luck, and I'll see you sometime. Bye-bye.